The first thing I want to say something about is DNA and chromosomes. And I want to make a sort of fuss about this because people I know have a hard time connecting chromosomes, those popsicle sticks you guys work with in lab way back when, with the big A's and the little a's. And what I want to try to do is connect that now to something else. So I want to make sort of a fuss about how that works to get people thinking about it, because it's sort of a bit of orientation. And so if you guys modeled a chromosome, you know, with a popsicle stick and some gene on it, right? Let's just say a chromosome. So you model as a popsicle stick. What's the actual detail? What were you looking at? If you zoom in, if you zoomed in, you'd see, I'm terrible at drawing helices, but I think it's, yeah, that's not too bad. All right, it's this double helix thing. It's this DNA. So those chromosomes are DNA. We will write it as, because it's hard to draw it in that double helical way, I'm going to write it in a flat projection. But you have two backbones. All right? And then you have, so there's another backbone the other way. Right? And then you have, and you guys saw this on the video and all that we looked at, there are bases sticking out of each of the backbones facing each other. They're bases. And between the bases are base pairing via our old friend hydrogen bonds. Okay. So that's the structure you guys all saw. There's a note I want to make about this. So, therefore, a chromosome, oftentimes people will say it is one DNA molecule. It's a shorthand. People say a DNA molecule, a chromosome. Is that really correct? If you take, if a molecule is a covalently bonded unit, there are actually two covalently bonded units there. There's one strand covalently bonded to itself and another strand covalently bonded to itself. And those two strands are not held together by covalent bonds. They're held together by hydrogen bonds. So strictly speaking, this is really two separate DNA, two separate single DNA strands. Each is a separate molecule. So it's really two molecules of DNA linked by H bonds. Okay. So sometimes you'll hear this, a sort of DNA molecule, and it can be confusing because a chromosome, a double strand, all those molecules that you saw in the warm-up quiz and in the book, they always had two strands. There's no covalent bonds connecting one strand to the other. It's only hydrogen bonds. So strictly speaking, they are two separate molecules. So so sometimes you may hear that shorthand, a chromosome is a molecule of DNA. It's better to say it's two strands, two hydrogen bonded strands. In any case, that's what a chromosome looks like. Because one of the ways to goof this up is people might think that those two blue strands, if you're diploid, one's big A and one's little a, and that's not true. A big A allele is actually a double-stranded piece of DNA like that. So we'll talk about more examples as we come to that. But that's one little piece of orientation. What I want to talk about today is, this is an animation that one of you guys made that I thought showed this particularly well. We want to talk about this process. So you've got chromosomes, right? So this is a double-stranded DNA molecule, another double-stranded DNA molecule. To be careful, there's one strand and another strand, and they're hydrogen bonded to each other. And there's one strand and another strand hydrogen bonded to each other, etc. So up here, there are actually 8 DNA molecules. Each of these represents a pair of things hydrogen bonded to each other. And so what I want to talk about today is how these guys replicate. So you guys animated, I thought this was kind of a cute animation. They like sort of fade into 2, right? And then they X each other. So like they start as 1, and then poofo, there's 2, right? So what we're talking about today is that process. Does it appear like that? No, that's what you have to do if you're animating with popsicle sticks. In real life, how does it actually happen? And so that's the process today that we're talking about. And the goal is to copy the whole chromosome. Right? In fact, all the chromosomes you got. So let's take that piece by piece. To understand how that works, there's a bunch of really simple rules that will apply all over the place for working with DNA and RNA. And there's a lot of things you could just sort of memorize, like certain words will come to them. It's actually much safer to remember these rules. These are prime cheat sheet candidates, right? And you can apply them in all the situations we're going to talk about in molecular biology. There are very few things in biology that have nice, solid rules like these. It turns out in DNA and RNA, there's some nice, simple rules that apply across the board. Of course, there are exceptions, but we will not sweat about them in Bio 111. Number 1, DNA and RNA polymers have a direction. We write it as 5' to 3'. And you guys saw that in the quiz. It's analogous to amino and carboxyl in proteins. It's analogous to that train of elephants. You have a tail end. If they're always linked trunk to tail, trunk to tail, trunk to tail, you have a free trunk end and a free tail end. 5' and 3' we'll talk about next lecture on Wednesday. Where they actually chemically come from, but they're just chemical names for those. And as you saw in the quiz, most people got this one right, the sugar has a particular shape to it, and one part's the 5' end and one part's the 3' end. Therefore, one end is different from the other. Okay? Like amino and carboxyl in a protein. And so we can write it in several different ways. You can say the 5' end, all right, and you can write it as P. You can draw it this way if you like. This is the sort of most detail that we'd look at. And I'll show these piece by piece. A and T, for example. So this is the backbone, this running along here, and it consists of phosphate and sugar. All right? And these guys are the bases. And you guys saw this on the quiz. And again, this is the sort of extreme cleverness of a backbone side chain model. What makes these things cool is the backbone is the same no matter what the base is. So I can link DNA in any sequence I like. I can put as many of them together in a line as I like, and I can put A, G, C, T, A, C, G, T, A, whatever I like, right? Because the backbone where it connects is the same. It does not matter. The bases just hang off the side. So that's the sort of cleverness of this bit of evolution, is that the backbone's the same, so you can link them up any way you like, and the bases are what cause the difference. We can simplify it. Sometimes it's useful to write things like this, 5' to 3', A, T, just to sort of draw the backbone as a very schematic sort of a thing. Or finally, 5', A, T, 3'. Right? Just where all we really care about is that abbreviation. And you could, if you really wanted to, go from that to write out any of these other structures, or even the full-on molecular structure, all the carbons and hydrogens and nitrogens and all that. We will never worry about that in Bio 111, but that is enough to specify its complete chemical structure. Questions about that little bit there? 5' and 3', the backbone bases, that sort of stuff. Cool. Next, this was not in the book as clearly, and so I want to make a fuss about this here. Polymerization that is adding bases, is always goes 5 prime to 3 prime. That is, you add to the 3 prime end only. Okay? So, all right, in the real world, there are rare exceptions to this, but this is one of those few rules that has very, very few exceptions. So if you're going to grow a new strand, you're always adding to the 3 prime end. That's an important rule. You can't grow in either direction. There's only one direction. So on that strand, if I wanted to make that strand longer, I can make it longer by adding bases to this end. But I cannot make it any longer by adding bases to that end. There are no enzymes that add bases going in that direction. So that's one thing that's important. Now that comes into play in a couple different places. The other thing is, and the new sequence is based on, that is, how do you know what the new bases are supposed to be? They're based on base pairing. Base pairing. And that's always, again, there are weird exceptions because this is biology, but we're not going to worry about them. A to T or A to U, we'll see some examples of that, and G to C. And these are by 2 H bonds, and this is by 3 H bonds. And you guys saw that as well. That's how you could, in fact, one of the ways you could identify the bases when you were supposed to figure out the sequence is whether there was if there were 2 hydrogen bonds, it was AT. If there were 3 hydrogen bonds, it was a GC. Base pairing is huge. There's 2 things I'll say about that. It's one of those things where if you fall asleep in lecture and you find me asking a question, how does this work in the next bunch of lectures, just say base pairing. You'll always be right. Turns out everything we're going to talk about works by base pairing. So again, remember that if you fall asleep, say base pairing and you'll be right. The second thing is, I was lucky enough many years ago to actually meet Jim Watson of Watson and Crick. And one of the things that's hard, knowing all of this, is what was it like back then when they figured out the sequence of DNA. What he said was sort of the cool part, the part that they figured out that then they knew it had to be right, was the sort of elegance of base pairing, the fact that it explained so many things that they knew then, and not only that, all the things it explained since. So base pairing is huge, right? It turns out to explain a whole bunch of things that at the time were very exciting when Watson and Crick discovered the structure, but was even, since then, is even more dramatic. All right, so there's that one. And then the last thing, which is a little less obvious, but is also very important, is that base paired strands must be what's called anti-parallel, which is a weird word, but basically it means parallel but run 5 prime to 3 prime in opposite directions. So, for example, 5 prime, and we'll usually write them with an arrow going that way because that's the way the strand would be made. 5 prime to 3 prime. This would be okay. This is okay, but not 5 prime to 3 prime. All right. So this guy, never. You can never find that. All right. So why is it this way? You can't see that until you actually build a 3D model of this, and the things just basically don't fit in 3D. It's totally not obvious from 2D. And so you just have to take my word for it. They have to be anti-parallel, go in opposite directions. That's just the way it is. Turns out there are chemical reasons for that. Right? Questions about those rules before we get down to business using them? Yeah, sir. Oh, would it be wrong? Would it be wrong if these guys went all the way across? No, I was just being careless. They could go all the way across, right? Because they indicate the base pairs, and they really should actually go all the way across. I was doing because the diagram looks a little better drawn like that, but they do in fact go all the way across. All right, other things. Yeah.
So the bottom, the 5 prime and 3 prime in the opposite direction will always be molecularly different.
So the bottom strand will always be molecularly different. It will be a separate molecule, But if you know the sequence of this one, you know the sequence of that one because they have to base pair. Right. Right. So they will be different molecules, but they will have predictable relationship between their sequence. All right. Very well. What I want to do now is talk about the DNA treasure hunt thing, which you guys just started, which is going to be a fun thing by the very end of class. So the DNA treasure. And this is legit in the sense that my friend, her name is Jo, let me write that neatly, Jo Handelsman. I always spell her name wrong. Handelsman's lab found this DNA in a soil bacterium. And what does it do? So they isolated it for a variety of different reasons. They found a number of genes that they could figure out what they did. But there were a bunch of genes they have no idea what they did. And so she gave it to us to see if we can find something cool. So we'll learn about how you find something cool in a random DNA sequence, and we'll see. Maybe you guys, your particular piece might turn out to have something interesting in it. And like I said, this is research. They actually figured out some of the genes, but this was a big region where they were like, no, we don't know. And so there's been a lot more information gathered since they published this sequence. And so there's a very good chance that we'll find something that is now known. And so that's why it's a treasure hunt. There's a good chance we'll turn out something kind of cool. So everybody gets a little bit of this. Is a huge, long piece of DNA. Each of you gets a little bit of it to focus on. And like I said, with some luck, some of you will turn up something interesting. So for the first phase, we're just going to talk about DNA replication. Did you have a question? Yeah. Yep. Mm-hmm. Yeah, so the question was about adding nucleotides. On the bottom strand, you add going this way. And on the top strand, you add going this way. That's exactly right. All right. So phase one is each of you, I hope, brought in your DNA sequence. So part one is DNA structure and replication. Right? And the goal is to copy all of both strands. Okay? So we're going to take this step, and each step what I'm each step what I'm going to ask people to do is write on a separate piece of paper, because if your name comes up on the student picking application, I'm going to want to grab it and put it on the projector up here and show it to people so we can think about it. Right? So today some more trees will be sacrificed than on other days. But remember, if you work in groups, you can share that piece of paper, so you don't all have to do it. So we're going to take it step by step, and since you all will have several DNA sequences in your group, it's probably in a group better to just pick one, because it won't matter which one you use. They're all the same from this point of view. So number one is redraw your DNA with the first about 5 base pairs, unzipped. Okay? And the remainder still paired. And use a diagram like this. Something like 5 prime C G C T da 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 3 prime A G C and then G whatever. So these are the base paired parts, and that. So this is unpaired and paired. All right, and that's a 5 prime and that's a 3 prime. So the first thing you do is pick one of your group's DNA sequences and just rewrite it with the first couple, copy it down, and rewrite it with the first couple base pairs, first 5 or so base pairs unpaired, because we're going to start replicating this thing. And so the way you start replication is you unzip it, as you guys saw in that animation. So redraw it with some unzipped, and then I'll give you a couple minutes to do that, and then I'm going to call a name.